method after this character. So please welcome Florian Wagner.
Okay, so thank you to the organizers for this amazing event, and thank you for the opportunity to present my research. I'm going to be talking about a machine learning framework that we've been developing to define cell types in single-cell RNA-Seq data called Moana. And so we all know that single-cell RNA-Seq is an extremely powerful tool to study complex tissues. And in the past, there have been all these papers describing tissue atlases. For example, I'm showing here an example from the mouse cell atlas by Han et al. This is for kidney. And so the authors identified all these different cell types, all these different cell populations, and they also did some in situ hybridization experiments to confirm the cell identity of those populations. So what happens when you do a single-cell RNA-Seq experiment on, let's say, mouse kidneys? How can you actually make use of this knowledge that was described in this atlas paper? And so you would like to transfer these labels essentially, or you want to make use of these cell type identifications. But right now, this is a very difficult problem because we don't really have quantitative definitions of cell types for single-cell RNA-Seq data. So we think it's very useful to think about this problem as a classification problem where, let's say you're the person developing this atlas, you're training a cell type classifier and publishing that, and then anybody who does an experiment on the same tissue can use that classifier to predict cell types. When you think about cell type classification, it turns out to be a very interesting problem because there's all these interesting challenges associated with it. So first of all, we know that cell types are related to each other, some of them more closely than others. So while it might be very easy, for example, to distinguish myeloid cells from lymphoid cells, it might be very difficult to distinguish different subtypes of T cells. So essentially, this is a multi-resolution problem, and any type of cell type classifier needs to operate at these different levels of resolution. Second of all, a cell type classifier is really only useful if you can use it in different contexts. So for example, if you're studying a disease model of kidney, it should be able to identify the cell types also in the disease state. Otherwise, it wouldn't be very useful. The same when you're doing some sort of experimental perturbation, the cell type classifier still needs to be able to recognize the cell types. And so finally, whenever you're working with single-cell RNA-Seq data, you have all these technical challenges to worry about, like the data sets are very large, very high dimensional, and most importantly, there's all this technical noise, which we refer to as sampling noise, owing to the fact that we're only capturing a small fraction of the transcripts in each cell. And I should note that I think right now we're still sort of in an early stage where there's not that many data sets to choose from, where you have like both a normal, let's say, and a disease state. This is also, I think, something to keep in mind. When we started working on this project, the only method that we were aware of that really does classification for single-cell RNA-Seq data was SEMF Cluster, published by Kiselev et al. And I'm simplifying here a little bit, but not too much. The way this works is you're selecting, for training, you're selecting variable genes. Then you're calculating average expression profiles for each cell type. And then for prediction, you're simply calculating correlations with these average profiles for the selected genes for each cell in your test data set. And so conceptually, there are some advantages to this. Especially the training is very straightforward. You maybe need to optimize one or two parameters, but it's very easy to do. And also the prediction is very fast. You're simply just calculating correlations. But there were some limitations that we recognized that we wanted to address. So first of all, the way SEMF clusters reduces the dimensionality is basically just through feature selection or gene selection. And when you think about the expression differences between different cell types, you realize that there are these large sets of correlated genes that are highly expressed in one cell type but not in the other. So ideally, we want to utilize this concept of gene modules and maybe define some latent variables that we can use to do the classification on. And second of all, it's not a hierarchical approach, and we think a hierarchical approach would be very useful considering this multi-resolution problem that I talked about earlier. So we wanted to address these limitations, and to do so, we focused on PBMCs as a model tissue. we think PBMCs are very useful for this type of problem as a model because we know a lot about PBMCs, we know what cell types are in them, and we have these different levels of resolution. So we have myeloid cells, lymphoid cells, different subtypes of T cells. So I wanted to describe very briefly how our framework works. So first, basically, we're just doing PCA, let's say on the entire PBMC data set, and then you get something like this. Then we do clustering and cell type annotation, which I'm not going to go into right now. But basically, you see that the first principal component separates out the myeloid cells, and the second principal component separates out the B cells. So this works pretty well. And then the idea is to just fit a linear SVM classifier on this PCA transformed data. And so now each principal component really represents the expression contributions of many genes. So it basically captures an expression module. And so this seems to work pretty well, but of course you're not done yet, because, for example, this cluster represents both T cells and NK cells. But what you can do is you can repeat the same procedure again, just for the T and NK cells. The problem is that you're actually not getting these clear clusters anymore when you do PCA. And we think that, however, we did some clustering analysis. It turns out that the first principal component does actually capture the T cell kind of expression module to an extent, but there's no clear separation. And we think that this is not because there's some sort of continuous biological phenotype. We think this is basically just due to technical noise. Fortunately, before that, we developed an algorithm called KNN smoothing for removing noise, technical noise from single-cell RNA-Seq data. So when you apply KNN smoothing and then do PCA, you get something like this. So here, I don't know if you can see this, but on the top right side here, right here, there are the NK cells. They're now forming like this isolated island, and these cells down here are actually different types of T cell populations. So now we can separate the NK cells very easily in this PC space, and we can fit in a linear SVM to that. So that's the basic idea. I want to say that for clustering, we're actually also using KNN smoothing, and this is actually what motivated us to design this framework in this way, because we generally observed that after smoothing, you do a much better job distinguishing different subpopulations in PC space with just one or two PCs. And so, for example, you can look at the smooth expression profile of a T cell marker like CD3D, and you see that all these cells express very high levels of the this T cell receptor gene, and the NK cells express no T cell receptor gene, which makes sense because NK cells don't have a T cell receptor. Okay, so because we're doing this hierarchical approach, we're taking this hierarchical approach, it actually turns out that we can construct an entire classifier from a single large data set without ever analyzing the entire data set. So the way this works is you just sample a few cells from the entire data set. You do this, what I just described, clustering and training a classifier, and then you're using the classifier to predict the cell type identities of all the cells in the data set. And you're taking each subpopulation, and then you're recursively doing this procedure over and over until you've separated all the cell types. So this is very important because with data sets containing tens of thousands of cells, it becomes kind of very computationally inefficient to train SVM classifiers and to do clustering. So we did this, and we built this classifier that distinguishes between 9 immune cell types, 5 cell types listed here, and then 4 different types of T cell subtypes. And in our preprint, we show that this works pretty accurately if you're testing the performance on a normal PBMC data set. But what I wanted to focus on here was an application to an experiment where there's actually a treatment effect, where this is the more interesting case where you have a control and a treatment. And so we applied this to a data set published by Kang et al. So here we have 2 samples, 1 control, 1 treatment, of PBMCs. And the treatment is PBMCs were exposed to the cytokine interferon beta for 4 hours. And you see that when you put both data sets together into a t-SNE, they separate by a condition. So probably some combination of batch effect and treatment effect here. So what we did is we trained a classifier on the control data set, and then we used it to predict the cell types in the treatment and also in the control data set. And what you get is something like this. And you see that the classifier essentially completely ignores whatever batch effect or treatment effect there is, and it manages to assign cell types independently of that effect. So we compared these results to a previous analysis of the same data set by the Sajija lab. So they described this integration approach, and they did some clustering, and we find that our results are in very good agreement with their cell type annotations. And I think it's also important to, at this point, to kind of compare these different approaches. So when you're doing integration, you basically have to analyze all of your data sets jointly, whereas when you're taking a classification approach, you can train a classifier on one data set, and then you keep doing experiments, let's say, and you can just apply the classifier to each data set as it comes in, essentially. You're not forced anymore to analyze all the data sets in a single analysis. So in the last part of my talk, I wanted to briefly talk about some unpublished work. So, let's skip that. So here we're looking at a kidney tumor. So this is clear cell renal carcinoma, a data set from Young et al., published in Science. And what's interesting about this data set is that the cancer cells actually only represent a small minority of the cells. Most of the cells here are either immune cells or some sort of stromal cells. So we wanted to identify immune cells in this data set. So we looked at the CD45 expression. So here we've just identified the clusters of cells that correspond to immune cells. And then we wanted to know what would happen if we just put all these cells, all these expression profiles, into our PBMC classifier. And we got some really interesting results where certain cell types were completely absent. So for example, there were no naive T cells, which makes sense, I guess. But then there were also some sort of interesting results. For example, these cells were classified, half of them were classified as CD14 monocytes, the other as CD16 monocytes. So that's interesting. Of course, you have to take these results with a grain of salt, because the classifier will always classify cells as something. It will never reject it, but this could provide some hints. Maybe these are macrophages, not monocytes, but it's an interesting tool. And more generally, I think what this suggests is that maybe it's possible to train an immune cell classifier that's able to identify immune cell types across tumors, so that you would have a quantitative basis for defining all these different immune cell subsets across tumors. And that, of course, would be very useful in the context of studying immunotherapies. Okay. So I hope I've shown that our framework allows you to quantitatively define cell types in single-cell RNA-Seq data, that you can predict cell types across experiments and conditions, and that in this particular example, at least, it could help identify immune cell types in kidney cancer. So you can find our preprint and the Python implementation here, and if anybody is so proficient in Python and interested in using this, just let me know, because right now we're sort of developing the documentation for this. So I want to thank my advisor, Itai, who's been very supportive of me for the last 2 years, and ever since I've met him, which is almost 10 years ago. And I want to take your questions. Thank you.
Is it? Okay. To what extent that smoothing actually affects the properties of the classifier? Because you clearly change the variance there and so on.
Yeah, that's a pretty broad question. I mean, in general, the way I think about it is smoothing basically just aggregates expression profiles. So you have to be careful about how you normalize things. But otherwise, other than that, the main effect it has, I think, is that you're able to suddenly really clearly separate cell types in PCA space, whereas before, it's very difficult. The principal components don't really clearly separate between the cell types. So I think, the way I think about smoothing generally is if you're not over-smoothing, you're actually recapitulating the original expression profile of the cell much, much better than if you were to look at the raw data.
So my question also for the smoothing, so how do you choose K, actually? Yeah.
Yeah, so it depends. We're actually using smoothing in two different roles in this framework. One, we're using it for clustering, and then we're also using it for classification. For clustering, we're smoothing really aggressively sometimes, because that really helps us to much more easily just define these clusters, and we're basically forcing cells to be in separate clusters. But for classification, we're trying to smooth as little as necessary, because it affects like how much. The more smoothing you do, the more you're risking to over-smooth the data. So you need to have larger sample sizes and all that. So we're trying to use as little smoothing as possible. So the smallest K is such that we're getting a good separation of cell types in PCA spaces.
So the K, choosing of K doesn't affect the data shape, or say the cell. So you don't get a you don't get a cell clustering like false kind of positive, like based on this. Once if the noise, for example. Yeah, if there's noise and
you can definitely do harm if you're doing too much smoothing. We're working with relatively large data sets, so that we're generally trying to stay below maybe 5% or even less than of the total number of cells.
I see. Also for the classification, I think you do this iteratively. Is that the understanding correct?
Yeah, we're doing it like kind of hierarchically. Yeah.
Okay. So when do you stop?
We're stopping when we're not able to just identify sort of biologically meaningful subpopulations, essentially. Like we look at genes and gene expressions, and when there's no more clearly defined clusters, we stop it.
Okay, that's clear. Thank you.
Okay. So within the iterative procedure, within the recursive procedure, which metric do you use to establish the optimal number of cluster at each step?
So that's an interesting question. It doesn't really matter that much, because it's essentially up to you. So you can decide that you only want to distinguish it in each step, which is what we've actually done initially. We've only just identified clusters in each step. Oh, thank you. Sorry. So I'll repeat what I just said. So it's actually up to you how many clusters you choose in each step, because you can decide that you only want to define 2 clusters and then keep going with each of the 2 clusters, or that's what we've done here. But you can also try to identify, like we typically limit it to 2 or 3, because in 2-dimensional PCA space, it's often very difficult to distinguish between more than 3 populations.
But then it's not optimal, right? If you get to choose at each step how many clusters you need, and then go manually through each step, then it's not automatic.
Right. It's manual essentially. Yeah. It's not. There's no automatic procedure for that. But maybe that would be interesting to do. Yeah.
Thank you.
One last question.
So your KNN smoothing thing reminds me of this mutation-based approach from MAGIC. I was wondering if you would comment on that, and if you've compared it and how it affects the performance. Do you know about the MAGIC method?
Yeah, I know the MAGIC paper. It's quite different. I think conceptually the main difference between KNN smoothing and MAGIC is that the smooth profiles, the way smoothing works with KNN smoothing is that you take an unweighted sum essentially, or an average of a certain number of cells, whereas in MAGIC you have weights put on each cell. So MAGIC calculates a weighted average of profiles, and also the way MAGIC identifies which cells to use for smoothing is very different from the way we're doing it. So there's